Hello folks, we are ready to do part 9 of neurophysiology, and I'm going to talk about how our synapses work. A lot of this is going to be remedial, but hopefully we'll answer some questions that you have. So first of all, let's define our synapse. Okay, this is where two structures communicate. Okay, so for a synapse, you're dealing with the movement of charged ions. So typically we're dealing with some kind of neurotransmitter, but there are examples where you use a different type of stimulus. But for now, we're just going to go with that. So right here, key idea, you're going to transmit information from one structure to another. This could be from a neuron to a neuron, or it could be from a neuron to a muscle cell or a neuron to a gland. Now, when we're dealing with this, I want you to focus on pre versus post. So you've got a neuron, the one that's sending is going to be presynaptic. The one that is receiving is going to be called the postsynaptic neuron or the postsynaptic structure, okay? So if we look down here at this particular example with our spinal cord, we have in blue a stimulus that's going to go along a sensory neuron, and then it's going to synapse right here. In this case, this is presynaptic, and the green is postsynaptic. But we're now going to have the green interneuron communicate with the red neuron. So now this one is going to be the presynaptic, and this one is going to be postsynaptic. Okay, and this is basically the way it's just going to work along all of these. Okay, next slide. All right, for most of what we're dealing with chemical, that wasn't supposed to happen, chemical synapses, which means the chemical is going to be a neurotransmitter of some sort. The one that we're most familiar with right now is acetylcholine, but there are definitely others. This is making me irritated the way it's kind of not even. That's a little better. All right, so what I like about this picture right here is I've got the cell body of my neuron, and then I've got all of these dendrites right here. Oops, that's not one. That's one right there. And then I've got my axon right here. Then that's all postsynaptic. So this is all postsynaptic. I have a lot of presynaptic structures here. So this is a presynaptic neuron. Here's another one. Here's some more. So these things with the right here, those are axonal terminals of a presynaptic neuron. Now we've got some cute ones here that are green. All right, we've got some more right here, etc., etc. I'm not going to trace all of these, although it's kind of relaxing to trace them all. So those are all the presynaptic neurons that are coming up to communicate with the dendrite. All right, I'm going to erase some of this now so that we can see what's going on with these. Okay, so where you see the red, and we're going to get into this in I think it's the next lecture, but in a minute. Red is inhibitory. These are the IPSPs, so inhibitory postsynaptic potentials. And then green is excitatory, so excitatory postsynaptic potentials, okay? So it changes what neurotransmitter you're using in order to stimulate them, okay? Don't forget, with synapses, they can be axosomatic. So that's the axon synapsing with the soma or cell body, okay? So this would be an example of axosomatic. Or you can have axodendritic. These would be examples of axodendritic synapses. And then you have a few axonals. And I don't really have a good one for that. If I had one right here, that would be an example of axonal. Okay? All right, let's look at the next picture. All right, actually we're doing a Blackboard slide now. So the two major types of synapses that we've so far been dealing with in this course are electrical synapses and chemical synapses. The electrical, the place where you've seen this is with gap junctions between muscle cells, okay? This allows the movement of charged ions and other things as well, you know, nutrients, gases, to move between cells rapidly. All right, if it's charged ions, it could lead to a local potential or action potential. If it's gases, it might be oxygen, like with our bone cells, okay? This becomes really important for synchronization between structures. You can have gap junctions between neurons, although in this course, mostly we've seen the gap junctions between muscle cells. All right, the chemical synapses, they are all about the neurotransmitter, okay? So the neurotransmitter is the chemical. And you could be dealing with acetylcholine, dopamine, GABA, etc., all kinds of things. And their job is to open chemical gates, which are also called ligand gates. All right, when they open the gates, then sodium or other charged ions can enter. Okay? Now, something to keep in mind is directionality. When you're dealing with an action potential or a local potential, there are several mechanisms that keeps things going in one direction. One of those things is the refractory structure. So the refractory period, if you remember that, the gates, whether they're voltage gates or chemical gates or the sliding filament mechanism or all of these things, they have to reset. So a signal can't flow backwards because those structures are resetting, okay? Let's see. Hold on just a second. All right, let me see, what do I want to do? Let's kind of go over that chemical synapse again, because I think this is a good activity for you to practice. So this would be your presynaptic neuron. Okay, and then I'm going to come down here and I'm going to kind of draw a postsynaptic neuron. This could also be a sarcolemma if you wanted it to be. Okay. So if I change the color here, you got an action potential that comes down here. It's going to come to a calcium voltage gate. And then calcium enters here. And then I have vesicles with a neurotransmitter in them. Okay? The neurotransmitter is going to come out here and it's going to float to the gate. And then the gate is going to open and sodium is going to enter into this area where it can depolarize locally. And if you remember how far the signal goes depends on strength of stimulus. If a lot of sodium enters, it'll be a strong signal. If not a lot of sodium enters, it'll fizzle out. But if it's strong enough to get over here where I have voltage gates, so it needs to get over here and reach threshold. If it does that, you will then propagate an action potential. Okay? And then it's going to just go along until it runs out of voltage gates. All right? So this should be review at this point in time. We've done this pathway several times with the muscular system and with the neurophysiology. So I think you should be pretty good on this concept, and I've drawn it out for you several times. you should be drawing it out for yourself as well. So I have another picture right here, which is prettier than my drawings. So you can use this to help work your way through those steps. So you can see the action potential is coming down here. It opens up voltage gates, calcium comes in. The calcium is going to trigger the exocytosis of these vesicles. Acetylcholine or GABA or dopamine neurotransmitter is going to come over here. It's going to open the chemical gates, and then sodium is going to go through the gates and depolarize the membrane. If it is a strong enough signal that it gets over to here and it's at threshold, you could generate an action potential. If it doesn't, then it's just going to fizzle out. Okay? All right, I think that's it for the slides that go with this neurophys part. So I'm going to go ahead and stop the recording.